Let's talk about something people always ask me. How do neuroscientists study the brain without actually looking at the brain? Well, here comes my reality. We grow tiny little brain-like structures called organoids in the lab. No, they don't think, they don't feel, but they're very useful in research. So let me show you what organoids are and why we use them. So, what even are organoids? Organoids are tiny 3D clusters of cells that organize themselves into structures resembling real human organs. Think of them as miniature, simplified models, not the full organ, but a small, functional screenshot of it. When we make organoids to study human development, they form layers and patterns that resemble the developing human brain. They don't think, they don't feel, and they definitely can't help me with my thesis. But they do mimic human brain development in a way we simply couldn't do before. They're powerful because they behave like human tissue, giving us insights into development, disease, and cell behavior without needing a living human or animal model. The process may sound intimidating, but here's the digestible version. Everything starts with stem cells, and these are amazing. These are cells that can turn into almost any cell in the body. With the right growth factors, signals, and environment, they begin arranging themselves into clusters. So over days, weeks, months, and sometimes even years, these clusters start forming recognizable brain regions. It's a mix of science and patience. My favorite part is watching them grow. It's like caring for extremely needy plant babies, except these are way more delicate and they're way more expensive. Organoids let us explore biological questions we simply can't answer in humans or animals. They help us understand how certain gene mutations can disrupt brain development, why specific neurons fail to connect correctly, how early development processes differ between individuals, and potential treatments that can work in humans. These are especially valuable in studying neurodevelopmental conditions like autism, epilepsy, schizophrenia, or any genetic syndrome, places where timing, early cell identity, and connectivity really matter. With organoids, we can observe these processes unfolding in real time. Now, here's the part that everyone leaves out. Organoids are not tiny brains in a dish. They don't think, they don't feel, they don't dream, or even care about deadlines. So here's a list of things they do lack. Blood vessels, meaning they can't grow indefinitely. Sensory input, meaning they can't see or hear because nothing comes in. The full and complete variety of brain cells. The complete wiring you'll find in a real human brain. Immune cells, hormones, and all the environmental cues that makes us human. And because of these limits, organoids can really only model certain stages of brain development, and mostly early ones. Understanding what organoids can't do is just as important as understanding what they can do, because it's what keeps the research grounded, ethical, and realistic as the field grows. Now, do not get me wrong, organoids are powerful tools, but let's talk about the limitations that scientists barely talk about. First, variability. Even if you treat each organoid the exact same way, organoids can grow very differently. Two batches can behave like two completely different personalities. This makes standardization and reproducibility a real scientific headache. Second, size. Without blood vessels, organoids can't really grow very big. The inner regions don't get enough nutrients, so they do struggle to mature beyond early stages of development. Third, they don't fully model the complexity of the human brain. They're missing immune cells, vascularization, and environmental cues that shape development. Don't get me wrong, they are incredible, but not complete. And finally, they take time, as in weeks, months to mature. So it's not exactly a quick experiment. However, these limitations do not make organoids less valuable. They help us understand how to use them realistically. So, despite their limitations, organoids have already led neuroscience forward in ways most models couldn't. One of the biggest breakthroughs was understanding how the Zika virus affects developing brains. Organoids let us know exactly how the virus stunts growth and disrupts neural development, something we can never ethically test in humans. Another major win? Drug testing. Organoids have been used to screen potential treatments for neurological diseases long before clinical trials. And these discoveries are just the beginning. Organoids are not just tools, they open up entirely new doors in neuroscience. So that's the world of brain organoids, tiny yet fascinating. If you want more videos that break down complex science into simple ways, hit subscribe, like, and comment which topic you want next.